So I'm going to do a brief series introduction, and then Dr. Nussbaum will introduce you, and then you're free to go. All right, welcome everyone tonight to our Timothy A. Johnson Medical Scholar Seminar Series. As I think most of you in the auditorium have heard, this is a seminar series that was established in honor of VTCSOM's first Associate Dean for Research, and he had a vision of really immersing our medical students in research from day one with the idea of fostering discoveries that were informed by patient care. And so you can see that embodied in one of our identities up here. So this is the VTCSOM identities, where we have system citizenship, so someone who's thinking about the impact on the patient of all of the factors that surround that patient. Lifelong master adaptive learner, so somebody who is always thinking about the right questions and trying to fill their own gaps in knowledge. And then the scientist physician, which is not necessarily a physician scientist first, but somebody who is bringing the mindset of a scientist to patient care. And so this series is about bringing in people who value the science, the act of inquiry, and are using their own interactions with patients to inform that discovery process. So this is the last seminar of this year's series. We are in the process of building out our slate of speakers for next year. So for those of you online and those of you in the room, if you have ideas of physicians who are doing research and you're interested in having them come here, please nominate them. We love to partner with our clinical departments and with our faculty who have collaborators at other places. So please think about nominating somebody who you would like to hear from next year. I'm really excited now to hand over our introduction of today's speaker to our Chair of Surgery, Dr. Nussbaum.
Thank you, Dr. LaConte, and thanks to everybody for joining us in person and online. I'm really thrilled to introduce our speaker. He's had a busy day today with everybody, and Dr. Alberti is truly a surgeon scientist coming here from the University of Chicago, where he holds the Sarah and Harold Lincoln Thompson Professorship of Surgery. He's the Executive Vice Chair of the Department of Surgery, the Vice Chair for Research for the Department of Surgery, and Associate Director of an NIH-funded Digestive Disease Research Center, CORE, at the University of Chicago. He's a fellow of the Institute of Molecular Engineering at the University of Chicago. He's been continuously R01 funded by the NIH for over 25 years, including through 2026, and has trained over 100 undergraduate, graduate, medical students, surgical research fellows in his laboratory. He currently holds 2 STTR grants from the NIH and is the founder and chief research officer of Covira Surgical, a company that produces antimicrobial compounds that prevent infection related to postoperative complications. He studies the molecular pathogenesis of infection-related surgical complications, such as sepsis, surgical site infections, and anastomotic leak. And he's past president of the Surgical Infection Society. I believe Dr. Alberti is going to tell you a little bit more about his background and how he got to where he is today. And so I'm going to turn it over to Dr. John Alberti, and thank you for being our Timothy Johnson speaker.
Thank you, Dr. Nussbaum. Dr. LeConte, thank you for both of you for the invitation to be here. It's quite an honor, and know that for those of you that are training here in both general surgery or as scientists, know that you're getting the best training that the United States has to offer. This is a little bit of my background. I was a Spanish major in college, which you'll hear a little bit about. Yeah. What happened? Oh, they didn't get the, are we okay? Nothing happened. We got to go to the, something's going on. Come on now. There it goes. Okay, good. Do you want to maybe get that back up to where we were? Yep. There we go. Sorry. Okay, no worries. Thank you. So, as it says up here, I was a Spanish major in college. I went to visit a cousin of mine who was studying to be a doctor in Mexico. And I went down there and never left. I never applied to any American medical schools. Ended up at that hospital, which was affiliated with the University of Chicago at the time. And that's how I ended up there. I spent a year in San Francisco doing a research fellowship, and then ended up, I don't know, addressing something that seemed to be important to the field of surgical research and surgical complications. And, you know, that's my story, and I'm sticking to it, as the saying goes. But, you know, one thing to look at in these humble beginnings is it doesn't take much except adhering to what you believe to be an important question, and I'll address that in a minute. So I had to read this book in college, Don Quixote. If you haven't read it, you should read it. It was published in 1605. It was considered the first modern novel of all Western literature. And Jeff Matthews, the chair at the University of Chicago, who knows Dr. Nussbaum very well, they worked together at the University of Cincinnati for many years, said you should read this book. I told him I was a big fan of Walter Isaacson, who's a historian and writes wonderful biographies. And he wrote this biography about Leonardo da Vinci. And he said, innovation requires creating a reality distortion field. Like reality distortion. Well, you know, obviously those of you that have read Don Quixote know he was quite delusional and quite distorted in his reality. And then I remember reading about this in a full chapter in this book that my mother gave me for Christmas, Steve Jobs by Walter Isaacson, who, again, he's a biographer who studied Steve Jobs, and he dedicated a whole chapter to write, innovation requires creating a reality distortion field. Now, I learned about Steve Jobs. I'm not a fan. He was a celebrity junkie, loved being on stage, and he was obsessed with Bob Dylan. He even dated Joan Baez, which is kind of shocking. Now, was he smart? I don't know that he was smart. He dropped out of college. He obviously was visionary and had an innovation aspect to him, but he certainly wasn't as smart as most of the people in this room. And yet he created something important and obviously very lucrative for himself. But innovation, original science that is transformative to a field, is weighted heavily in NIH grants, and it really is required to be a successful scientist, in my opinion. And here's a little video that I stole off of YouTube from E.O. Wilson, who studied ants his whole life. He's an evolutionary biologist at Harvard.
You may have heard the military dictum for the gathering armies: march to the sound of the guns. In science, the exact opposite is the case: march away from the sound of the guns. So Wilson's principle number 3, march away from the sound of the gun. Observe from a distance, but do not join the fray. Make a fray of your own.
So there's something about those people that were smart enough to march away from the sounds of the guns, that those people that really felt that there's something innovative in their thinking. You have to be a radical thinker. So I heard this: the body of work is derivative, when I was co-chair of the Committee on Appointments and Promotions at the University of Chicago during a tenure decision. Now, I didn't know what derivative meant. I mean, I kind of knew, but I didn't know it was how you insult a basic scientist. You call their work derivative. Well, everything is derivative. My work is derivative. But it's something that is not original, lacks creativity, and is simply copying or heavily borrowing ideas from something else that came before it. Well, yeah. Now, this person had 2 R01s, and she did not make tenure and was dismissed from the university, which may be why the University of Chicago gets this reputation as where fun comes to die. So there's this worry that there's this vanishing physician scientist or scientist physician, as Dr. LeConte said. And you're about 42 years old or 44 by the time you get your first R01. This means your work is independent and considered innovative, and you're an independent investigator. And that's old. But the most transformative science seeks to change thinking in a field. It involves creating elegant and novel methods. Its intent is not to solve a problem, but to rather advance an understanding of it. And I call this solution agnostic work. Most of us go into science, those of us that are physicians, because we don't like the politics necessarily of practicing medicine, but there's politics in everything, and that's maybe why Max Planck said science advances one funeral at a time, because he felt like even coming up with an idea that's a radical idea is challenging for many. So there's a difference, and I said this at lunch today, between a professional degree and an academic degree, and quite frankly, most people don't know the difference. There are doctors on television who say, I'm a board-certified gastroenterologist, and I'll tell you what's good for your microbiome. I'm like, you look them up and they don't know anything. They've done no science. And we're seeing this even play out in our national politics. The people that are being put in charge are not the people that have been in the trenches necessarily. So you go to college, medical school, residency, you spend a lot of time, you're 40 and you feel like it's now me time. And your choices are create original science and have knowledgeable reviewers in the field judge, criticize, and reject your grants or papers. And believe me, rejection is the rule. Or you can be a master proceduralist, educator, cardiologist, dermatologist, work in a market that embraces your skill set, have meaningful work-life balance, and pay off your debt, take care of your family. I think the choice is pretty easy. So Richard Feynman, who's one of my heroes, was a physicist who won the Nobel Prize for discovering quantum electrodynamics. He said, science is like sex. Sometimes something useful comes out, but that's not the reason we're doing it. And he wrote this book, which is really a wonderful book, called The Pleasure of Finding Things Out. And you know, science, whether you believe it or not, is fun, because figuring things out is better than making stuff up. And that's one of the reasons to do it, in my humble opinion. So, in my opinion, the diminishing physician scientist is a product of natural selection. And I know Dr. LeConte and others are trying to make that happen. You're trying to make that happen here at Virginia Tech because it's important that we speak to each other, understand each other, and work together. And scientific research is a full-time job. It requires long training, college, PhD, MD, postdoctoral fellowships, repeated submission of grants, and this is most important, the ability to bounce back from constant rejection. Not everybody can do that. And it takes a special person to do that, and it's a problem. Clinical care in the operating room are seductive. You get paid a lot of money, you're respected, you're productive, you have an audience. In England, they call the operating room the operating theater. And you know this, Mike, they'll say, well, I'm in theater that day, I can't come, I'm in theater. And I'm like, well, what's that, the operating room? I'm in theater. And I'm like, it is kind of a theater. There's a performance, there's an audience, everybody's clapping, you feel good about what you did. It really is theater in its own way. So the formula to obtain, because people ask me this question, how do you ask me today many times during my constant interaction with everybody, how do you get continuous and sustained NIH or NSF funding? In my opinion, there's no formula. So here's my formula. Generate compelling preliminary data that provide clear mechanistic plausibility to a scientific premise transformative to the field. So let's break that down. Compelling preliminary data. I call this killer preliminary data. Who's going to give you the time and the money to do this? Who's going to hire you and say, okay, here's $1 million now for the next 3 years, generate killer preliminary data that is going to get you an NIH grant? You're like, whoa, that bar is really high. Clear mechanistic plausibility. This is the hard part. You got to knock a gene out or create a mutant or do something to prove causality. Can't be coincidence, association, causation, linked to. None of that works. It's got to be causally linked. And the scientific premise has to be transformative to the field. In other words, it can't be an incremental finding. It's got to these days, it's got to be transformative. Why is that? Because I know this from my own experience, 100 grants go into a study section. 50 are immediately triaged out. They don't even discuss them. They don't even review them. Well, they review them, but they don't actually discuss them at study section. The other 50, if you don't score in the top 10%, you're not getting money. So wait a minute, that's top 10% of 50s, but it's really 5% of the entire 100, because 50 are thrown out, not thrown out, but triaged. So it's pretty competitive, and it's getting worse. And so if you don't do something that's transformative, in my humble opinion, having served on study section for many years, it's going to be hard to get funded. So maybe today's lesson is, I really can't explain why I'm here in front of you. Somehow the system has tolerated my own distorted reality. In my humble opinion, there's no formula for sustained extramural funding success. It can't be prescribed. People came up to me earlier today, and said, how do I get funded? I'm like, I don't know. How do you write a hit song? I don't know. Passion, curiosity, credentials. Some people believe if you get enough degrees, you can do it. It won't do it. None of this guarantees success. And I will show you what I think does it. Now, there's publishable research, fundable research, and important research. And no, they are not the same. And it takes a lot of diligence to hang in there. Now, I was once told by a mentor, and this was when I was in California at UCSF, he said, there are 3 reasons to do research. And he was an immunologist. And I said, what are they, sir? And he said, travel, alcohol, and food. And I'm like, I don't think that's the reason to do it, but maybe. And this sounds immodest, but I've been everywhere, everywhere around the globe. And I will tell you this, it's nice to be wined and dined, but Spain has the best table food. They have the best table wine and the best table food so far. And, you want to understand why you want to do this. It shouldn't be about travel, alcohol, and food. It should be, I want to pursue this line of inquiry for the pleasure of finding things out and maybe being the first to know something that nobody else in the world knows. So let's get on to some science. When I started, the whole world was focused on how do bacteria trigger host cells. And part of it was this guy, who's a University of Chicago graduate, won the Nobel Prize in physiology and medicine for discovering that lipopolysaccharide binds to the TLR4 receptor and transduces a bunch of downstream genes. And it was very important work and transformative to the field. And he dissected out a lot of the molecular details of that interaction. And I asked a question that turned out to be very important also. Now, he was an MD, not a PhD. He did a neurology fellowship and decided to be a basic researcher because his dad was one. Now, I asked a question that turned out to be transformative to the field. I remember meeting with this brilliant molecular geneticist who was at the University of Chicago who discovered how bacteria behave in social groups. And we had a good connection. And we would sit down and have 3-hour lunches and write on napkins, and that's what it took. And I said, I want to know, since I'm a surgeon, if things go in the other direction, because we're beating up cells all the time. We're radiating patients, giving them antibiotics, doing some terrible things to people. I wonder if host cells, when they're stressed, activate bacteria in some kind of inter-kingdom signal exchange. Is it bidirectional? You're probably shocked that nobody asked that question in 1992. I don't know why I did. And then it involved the quorum sensing signaling system, which is interspecies and intraspecies signal exchange. And that's the title of this talk, which is you end up with a matchless web of dense dynamic interactions. It's not simple. There are like several moving targets in there. And this is why, correct me if I'm wrong, that I don't think you're going to make wine or cheese in the future by pouring wine down an HPLC or some mass spec or something and getting all the components of it, remixing it together and like, look, it's a $1,000 bottle of wine. Why? Why is that? Well, because you got a lot of bugs and worms and stuff in the soil, and they make this plant, and you got to have the sun and the right climate and the wind and I don't know, whatever, weather. And then you got to get the grapes and you got to smash the grapes properly, put them in these barrels, and these barrels have to have the right, I don't know, cracks and interstices in them and bugs in there, and it's complicated. And so I like to think of virulence or harmfulness or clinical infection as neither a property of the pathogen nor that of the host, but rather a property of their interaction. And how do you measure that interaction, or what's now called the interactome? This is where all this multi-omics stuff comes in. How do you measure something? We do what's called dual RNA-seq, where you can take bacterial or microbial cells and host cells, mix them all together, sequence everything and annotate and do contigs later to determine what is microbial and what is a host gene. And it's that interactome, or we think the metabolome, that really drives clinical disease phenotypes. And it's called quantum entanglement in physics. It's a phenomenon that occurs when two or more particles become so connected that they can't act independently. This may be, you probably studied this in physics, this Heisenberg uncertainty principle, where you can't just pull an electron out of its orbit and measure it, because now you're not measuring the thing you want to measure, which is, I guess, why they have to build these things in Europe. The Hadron beam cost $5 billion, and the Europeans built it on tax dollars, and they discovered the Higgs boson particle. Well, what practical use is that? Okay, now we know about the Higgs boson. So what? Well, obviously it was important to somebody. And for those of you that study cancer, well, is cancer a property of the host genes. It's not a property of the host genes or the environment. It's a property of their interactions. And that's what's really hard. So this academic clinician scientist being an endangered species is true. And I think this problem needs to be solved. And all of you young people here have to solve it. I'll be gone. But just like the mechanism in infection, cancer, and disease, the mentor-mentee relationship is neither a property of person A or person B. It's a property of their interaction. And that's why dating services don't work. Now, I have a partner. He's, he was born in the United States. He's of Indian extraction. And he's a brilliant, first in his class in high school, went to Northwestern in the 6-year program, brilliant guy, trained at WashU, and he's been married twice. And the first wife was a doctor, same ethnic background. You'd say, oh, these two are perfect. Didn't work. Second one, same thing, doctor, smart, beautiful, the whole thing. You're like, this is the perfect match. Didn't work. Now, I'm not going to give you relationship advice because been married to the same wife for 43 years, so don't ask me why, okay? But there's something about chemistry that we don't understand, and it all gets down to chemistry. And the lesson I want to give you is, this is on you. This is not your chair to find you a mentor. This is not your section chief. Find me a mentor. Find me somebody to write my grants. Get me a PhD. Generate killer preliminary data. Here's some samples. Do multi-omics and generate me a hypothesis. Doesn't work. You have to enjoy working together, and you have to have good chemistry with the people that you choose. And I can't tell which PhD should hook up with what. I don't like to use the word hook up. My daughters yell at me when I say hook up. Who should be paired with an MD? What PhD should be there? What project you should work on? Can't tell you what that is. What I can tell you is it takes a long time and it involves a long period of courtship. And if you want to say, what did you do that allowed you to be funded your whole career? I'll say, I sat down with a brilliant scientist, talked to him about my ideas for a long time, and we developed good chemistry together. And he liked my project and thought it was valuable to the field. So my hypothesis is that the dynamic function of one's microbiome influences all postoperative infection-related complications, and I'll show you some of those data in a minute. And even bleeding, Bobby Keski, who got his PhD in my lab on this last cycle, he's finishing a trauma fellowship this year, and we're hoping to recruit him back to the U of C. And he showed that when you grow Pseudomonas aeruginosa up to a certain point and then take its supernatant, it actually changes the coagulation profile. How can a bacterial exoproduct, something it secretes, change the coagulation profile? Well, where is that Pseudomonas? Does it have to be in the blood? No. Could it be in your nose or in your gut? Yes. Do those metabolites spill into your bloodstream? Yes. Well, that's interesting. So maybe postoperative bleeding has something to do with the microbes that we select for in certain patients that harbor those microbes. Maybe. And it turns out that it's all about food. And Bobby here, who wrote this paper, tried to understand how do you judge whether a patient's microbiome, their gut microbiome, is ready for surgery? And he called it defining microbiome readiness. And it's not just, looking at diversity or what components of the microbiome you have in there, but it's also their function. What are they secreting at that moment in time? So think about the patient about to have a colon operation. They're up all night pooping because we give them stuff to make them poop and clean out their colon. They're taking oral antibiotics. They're scared to death that they're going to wake up in a lot of pain. You think they slept that night thinking, oh, Dr. Nussbaum's going to open me up and find cancer all over the place. We call that peek and shriek. Going to open and close and say, nothing I can do. That's scary. That would be scary for me or anybody in this room. And so that person is sleep deprived, anxious, and worried about pain and up all night pooping. And now we're going to operate on them. Is their microbiome ready? Did we get it in the best shape possible? Well, I sent him to a cardiologist and measured his albumin and this, not enough, I don't think. So Ben Shogan, who is a colorectal surgeon now, he spent 7 years in our program, went to the Mayo Clinic, did colorectal fellowship. He just 2 weeks ago learned that his R01 to study dietary prehab before surgery as a mechanism to reduce complications after colorectal surgery was funded. It was funded by the NCI. So he's an NIH R01 funded investigator, and he did this study when he was in my lab. He showed that signals released from the operative field, the actual operative tissues, bind to bacteria, and he studied Enterococcus and Pseudomonas, in a way that increases their adherence capacity and their ability to produce these collagenase enzymes, collagenylytic bacteria. And those collagenylytic bacteria can directly break down the anastomotic wound, or they can, and both, and they can drive MMP9s, which are proteases, to go from their inactive to their active form. And supraphysiologic protease activity is the hallmark of all abnormal healing. And once he disabled the collagenase gene in these strains that he introduced, the phenotype disappeared. So he proved causality between the collagenase gene in Enterococcus and the anastomotic leak in mice. These were rats. Now, for those of you that don't remember this, collagen strength is a function of remodeling, which is a continuous process of synthesis and degradation. So when you create a wound in a patient, either closing a wound, closing the intestine, whatever it is, the aorta, you depend on collagen strength to remodel itself over time. And it might take a couple of weeks, but it gets stronger and stronger, makes all these important disulfide bridges and other things. It's a protein, and it does it by breaking it down, synthesizing, break it down, synthesizing. So what happens if it gets disordered by a bacterium? Well, it could be trouble. So in GI surgery, the most common cause of a deep organ space infection is an anastomotic leak. And we were able to model that in mice and know that 100% of mice have this epithelial on their tissues. And this is what I call the within group difference. So in the placebo-treated group, all the anastomoses heal perfectly fine. It's really hard to make a mouse leak. You can just throw some stitches in it. They heal 100% of the time. But in the rodents that did leak, they all had collagenylytic or collagenase positive enterococci on their anastomoses, whereas the ones within the group that did not leak, that was 50%. They did not have that gene there. And most of the leaks were subclinical. They showed if it looks like they're stuck, the omentum is stuck on them, and they kind of self-sealed. But if you rip that open and look at it in the inside, you see a hole. And only 10% had what I call a clinical leak. Very small amount had a clinical leak, which means they had an abscess there, which means the surgeon has to do something, or they actually had peritonitis. Things leaked out and caused a chemical reaction in the peritoneum. And we do not differentiate between who has a clinical leak and who has a dehiscence, because we don't want to be bothered at 4 o'clock in the afternoon if it's all contained and the patient's fine. You call Dr. Nussbaum in the middle of the night when the patient comes in with pain and the CAT scan shows bubbles and free air and stuff. Well, he's got to do something, either drain it, give antibiotics, go to the operating room. Something has to be done. But what's actionable in this space, we don't know, because we only count, this is major selection bias, when the patient has symptoms. We only image when they have symptoms. So the environmental context is very important. Now, somebody in my lab, Sanjeev Hoju and this group from the Netherlands, did this study where they said, let's make mice eat a Western diet. Let's get them chubby like patients. And let's give them antibiotics like we do to patients. And let's make them NPO after midnight, nothing by mouth after midnight, just like we do to patients. And lo and behold, that increased the collagenolithic microbes in their anastomotic wound really a lot. Went from 10 to the 4 to 10 to the 8. That's like a 4 order of magnitude rise. And that resulted in clinically meaningful leaks in this mouse model to 50%. Now, for those of you that don't know it, mice eat mouse chow. It's no meat, high fiber, low fat, low carbs. Who eats that? Nobody. None of my patients. They don't eat mouse chow. And if they did, they might have less complications, maybe. So this is what I call the surgical site infection paradox. And I got this from Neil Hyman, who said this to me one day. He used to be the chief of colorectal surgery at the University of Chicago. He said, if direct bacterial contamination is the cause of most surgical site infections, how can you do this and not cause one all the time? I think of the first guy that did a colostomy, probably woke up the next morning thinking, oh my God, this person's gonna have like necrotizing fasciitis all over them. Nothing. It's so rare to get a complication from this. And think about what we do. We bring out a piece of bowel. This is like a blowhole when somebody has diverticulitis or an obstruction or something. And you bring out a blowhole and you're literally sewing poop to the wound. Never get an infection. I don't want to say never, but it's rare. Then you do this, create the greenhouse effect. Let's put a little hood on there, get the moisture coming up and get the sun coming down and really make it grow good. Never gets infected. But if you ask 100 infectious disease specialists, how do you get a wound infection? Well, somebody spit in the wound. You dropped your head in there. You didn't clean. You didn't change your gloves. You're dirty. No. In my opinion, not the case. So we came up with this idea of the Trojan horse hypothesis for surgical site infections, where we said maybe a bug gets inside of a neutrophil or a macrophage and it silently travels to the wound site where it releases its infectious payload. And you're like, well, that must occur directly in the bowel. Maybe not. Maybe it's indirect. I don't know. And Monica Krazelek, who's now also a colorectal surgeon, spent 2 years in my lab when she was a resident. And she did really all the hard work on this. And we got GFP labeled MRSA from one of the microbiologists who was working on Staph aureus. And it lit up like, a light bulb. And we gavaged it down, and then we gave them antibiotics and operated on them and created a wound. And these neutrophils, which we gated, were the ones that ended up in the wound. Now we said, gee, can we take these neutrophils and put them on media, and will they grow? Because why is the bacteria inside of a neutrophil? The neutrophil should kill the bacteria, or the bacteria should kill the neutrophil. They shouldn't coexist, but there must be some molecular tolerance, molecular détente, something going on here. And this model was recapitulated by another group that basically did the same study, only they did it in orthopedic surgery. And they put a rod in there, did the same thing we did, used the GFP MRSA, and they found that the rod would get infected, the prosthesis in the bone would get infected only if it traveled silently inside of a neutrophil. Now, you might say, well, if my grandmother goes and gets her hip fixed and it gets infected, that damn doctor spilled something, our nurse, somebody spilled something on there. I don't know that that's true. But today, you can vectorially track bacteria by sequencing them and maybe find out, if the sink or the nurse put the Staph aureus into the patient, or the patient put the Staph aureus on the sink or under the nurse's fingernails, or the doctor's, or the operating room. Okay. So finally, this was done by Sanjeev Hoju, who worked with Rich Hoden, actually, and he sent them to me from the MGH, and he's been in my lab for the last 10 years. And he did this study where he said, let's feed mice a Western diet or chow, and then let's give them antibiotics or not. And then let's create a back wound. So he did this to the mice first. Then he created a back wound and created a surgical injury to the back, put a stitch in there and bovied the wound. and then made sure that nothing was growing there, put a protective barrier on there, and made sure at the end, at the time of the closure, there were no bugs there. And then clinically assessed them and found out that the chow-fed animals had a very low infection rate. It was mostly staph. But when you gave them antibiotics, the staph went away, and now it was gram-negative bacteria that infected the wound, not the staph. Remember, this is circumstantial evidence. Well, staph is on the skin, staph is in the wound, it came from the skin. Really? I'm a Homo sapien, you're a Homo sapien. If somebody gets murdered right there, you did it. You're a Homo sapien. You were there. You can't use that argument, but it's used all the time. And it's a wrong argument, in my opinion. And so if you give them a Western diet and give them antibiotics, that's when you really see an ugly wound. And it looks like this. And it's mostly gram-negative bacteria because we gave antibiotics that killed all the staph. And what comes back? The gram-negative bacteria. Okay, all these guys that we think come from the gut. So the 16S rRNA and ASV analysis, that's amplicon sequence variance analysis, demonstrate that strains from the gut and the wound are genetically similar. You never say they're identical because even the sequencing folks, the geneticists tell me, no two strains are ever equal. There must be something different. You can't say that's exactly that strain or that isolate. And so to prove this one step further, we swabbed their backs after their wounds were closed with live bacteria. And we couldn't make these. Most of the mice did not get an infection, kind of like the colostomy patients. You close the wound, nothing bad happens. You swab externally. You're like, oh, this happened because somebody spit on the wound or dropped some stool in there. Can't get it infected. Okay, and this is Bobby's last paper, and I'm almost done. And this paper was published just recently in Nature Microbiology. And it's really an interesting paper. And it asked this question, which is an important question, which is understanding what I call within group differences. So we took genetically identical inbred mice and injected them with a known strain of Serratia marcescens, a gram-negative bacteria, and said, why is it that some live and some die? The within group, not between group difference, the within group. Why within the group? Everybody's treated the same. Some live and some die. There must be a reason. And it turned out to be in the microbiome. It turned out to be a metabolite secreted by the microbiota. And these metabolites, we found them out. What they do is in the normal microbiota, they produce indoles through tryptophan, dietary tryptophan, which is its substrate, and it activates the aryl hydrocarbon receptor and a bunch of important downstream genes in macrophages that then go ahead and clean up the wound. Serratia marcescens secretes a exoproduct, a secreted product, that prevents the normal microbiota from activating the macrophages. So a successful pathogen has to disarm the immune system. And this thing did it really interestingly, which allowed people to show that there must be opposing forces. So you'll say, anyone that gets COVID is going to die. No, some people do fine, other people die. And well, I'll tell you what happens. If they get the wrong COVID in the wrong patient with a TLR4 polymorphism, no, that's not it. That's not 100% deterministic. There's some insight into the mechanism, but it doesn't explain it fully. So I want to just finish up by saying the question you pose is likely to be the most important aspect of your research career. You are a physician and a trained scientist or a trained scientist with a PhD. If you want to be a practicing scientist, in my humble opinion, today especially, I don't know how I did it. That's why I don't know. Don't ask me. I don't know how I did it. It's a full-time job, and that's why it's so hard to be a practicing clinician and a full-time scientist. And I stole this line from this book about Elizabeth Blackburn. She discovered telomeres or telomerase. And she generated killer preliminary data, demonstrated their vital role in processing or preserving information in the genome, mechanistic plausibility. And that was transformative to the field of genetics. And she told this story in this book about how she went up to her advisor and said, what's this crap at the end of the chromosome? And what did her advisor say to her? Don't do that, that's stupid. It's that Nobel Prize. She's a biochemist from UCSF, won the Nobel Prize. Same with Jennifer Duode, and I had the pleasure of listening to her speak at the University of Chicago. She's an RNA biochemist. She goes to Berkeley. She goes to her advisor. How is it bacteria get this extra chromosomal plasmid and they incorporate it into their genome, and when they're sick of it, they just spit it out and cut and paste and put perfectly what they wanted. How do they do that? Don't do it. It'll kill your career. Nobel Prize. 30 years later, she wins the Nobel Prize. And it's going to be the way we cure diseases like sickle cell and other primarily genetic diseases. So here's E.O. Wilson again. He studied ants. He showed how ants behave in colonies and in social groups. And he said this: there are 3 elements of a successful career. Knowledge, preliminary data. You got to do the work to demonstrate mechanistic plausibility. It's not easy. So here's me in medical school. I'm here with my friends. The guy to my left, which screen right to you, is from El Salvador. The guy all the way to the right is a Cuban American. The guy all the way to the left is from Nicaragua. The guy, and he was the smartest guy in medical school. I'm sure he's doing something amazing now. That guy. But I was fortunate enough to ask to come back and give a commencement address. And that was a lot of fun. And I didn't realize it at the time, but training there was, you are in one here. And you can produce original composition and generate the right chemistry with really smart people. You have a chance, at least. But you got to be in the right place, the right time, and I think that was me. And so maybe it was inevitable for me. I don't know. I have a lot of trainees, and I don't know. I don't know how to pick a winner. I don't know who to fund. You're all 10 times smarter than I am in this room. And there's something else going on, and I think it's obsession and willingness to take the risk. So remain faithful to your calling. Thank you, Dr. Leconte and Dr. Nussbaum, for the privilege of being here with you, and I'd be happy to answer any questions if you have any.
Thanks for coming to talk to us. I think the stuff about the Trojan horse is interesting, and the thought that maybe some of the infections aren't our fault in the operating room. I was wondering if you've thought about, and if you could speak to how you reconcile that with the fact that when we look back at like Civil War times and that kind of stuff, before we had a real Western diet and people were eating more from nature, the prevalence of gangrene and all of these other things. And I know there was some stuff with some of that lead and other poisonings, but have you thought about
Sure.
kind of just in general how we have less infections today than we did back in the day before the diet and obviously...
Agreed. Yeah, no, there's no question about one thing. Surgery is safer than it's ever been. We have the lowest infection rate that we've ever had. Antibiotics are a good thing. Sterility is a good thing. And that's what's moved the needle from 30% down to about 5 or 10%. But the question today for you is, how do we go from 5 or 10% to 0? How do you get to 0? I got the answer: more of the same. More sterility, more antibiotics. Is 2 good enough? No, give 5, give 6. Give antifungals, antivirals, give them all. I don't think that's gonna work because of resistant organisms. So we're in this U-shaped curve, right? Like the agonist receptor interaction where, a super physiologic dose of an agonist is actually inhibitory. A little bit of an agonist transduces the signal you want. Too much, the cell tries to protect itself and downregulates it. So to what extent can we move the needle from where we are even lower? Is it more of the same, or is it something different? And I'm glad they're antibiotics. I use antibiotics. We all use antibiotics. But the question is, we have diseases of human progress now. We have inflammatory diseases, multiple sclerosis, SLE, rheumatoid arthritis. We have resistant organisms in patients, and our prophylaxis isn't working. I'm going to show tomorrow morning 2 studies that show that 65% of the cases of MRSA, that if you get staph infection after spinal surgery, that 65% of those organisms are resistant to the antibiotics you use for prophylaxis. 65%, well, there should have been more infections. Yeah, there should be, but maybe the antibiotics and the host is fighting it. I don't know. The sterility is good.
Hi, this has been a really excellent talk. Thank you so much for, oh, sorry, was there someone else who was, oh, okay. Yeah, this has been a really excellent talk. I had a question. So you were speaking about the importance of generating killer preliminary data. There's been this sort of recent rash of particularly psychological research that's been completely falsified, like academic dishonesty down to the core. How do you promote like risk-taking and research in an environment where you're punished when you're wrong?
Yeah, it's a good question. Obviously, I've worked with scientists. I remember the one PhD in my lab, she would always look at me and go, this is good enough to publish. She goes, not good enough. Has to be done in triplicate. She was from Moscow, so she had like a Russian accent, and she was tough. And she kept me in line. I mean, she would not let anything go, unless it was airtight. And so that's one of the risks you take. Like, I'm going to say this tomorrow at Surgical Grand Rounds. I consider like surgical M&M, it's a place where you learn from the ignorance of experts. And it's because there are things we don't know. Ignorance doesn't mean, it's not the pejorative ignorance, there are things we don't know and we guess at. Like, do physicists and mathematicians understand turbulence? No, they can't even mathematically derive it, but does a pilot know how to manage turbulence? Yeah, they do. So you need to manage certain things. And we do that well as clinicians. We know how to manage a patient who has an infection, take a patient back to the operating room, stop bleeding. We know how to manage that problem. Do we know the cause of it? No. What's the onus on you as a scientist? Well, you better show me, like with the Pseudomonas study that we're doing and showing that Pseudomonas can induce a coagulopathic state. You'd say, what's in that exoproduct of the Pseudomonas? It's not ready for publishing anywhere. You said you got a supernatant that causes a coagulopathy. What's in the supernatant? What molecules are there? Did you separate them out? Did you separate them using centrocones by molecular weight, by osmolality, by density of some sort? Did you run them through an HPLC or a mass spec or something and find out all the chemicals that are in there? And did you test each one of them one by one on the system that you're using? No, we haven't done any of that. So it's an observation that requires a ton of work. And you want a paper in Science? Figure out what it is and find out where it bound and block it. And knock the gene out that expresses it, let's say in platelets. And you knock the gene out and say it won't work anymore because its target is gone. That's hard. Science is hard. Taking care of patients is hard too, but I don't think it's as hard. Sorry about that, Mike.
Hi, great talk. Thanks for coming. I just had a question about using antibiotics. Obviously, we don't want to promote the development of viruses and bacteria that can evade the antibiotic. And then, of course, there are bacteria and fungi in the body that you don't want to kill. So I'm just curious, how do you determine when is the right time to use an antibiotic and when it might be more appropriate to just let the patient's body do its job?
That's $1 million question. And, Rob Sawyer wrote this paper in the New England Journal of Medicine showing that 14 days of an antibiotic have the same outcome as 7 days. And it was called a stop it trial. And he said, why every time I operate on a patient with appendicitis or diverticulitis or something, infectious disease comes in and goes, keep the antibiotics going for 2 weeks. He goes, why is it 2 weeks? Is 1 week enough? And he found out, he did the study and published it in the New England Journal, and 7 days is enough. But why do we go 14 days? Well, if some is good, more is better. That's kind of how we think. Maybe that's right, maybe that's not right. But it's a hypothesis you have to test. You have to formally test it, and it took a lot of work for him to get that thing, 1, executed, 2, published. Took several runs. Who's going to do that? Who wants to do that? Hey, 14 days. What's it going to hurt? Maybe something. Maybe people get C. diff. Maybe people get fungal overgrowth like you were talking about. I don't know. But I think less is more in certain circumstances, and that we have to find that sweet spot. And the next pandemic is coming. I don't know who I spoke with, but then I said, is COVID a dress rehearsal for the next bad thing to come? Probably. And how are we going to fight it? With science. Somebody else had a question. Yes, sir.
Yeah, thank you for your talk. It was very entertaining, and I learned a lot. I'm just curious because you mentioned that part of the reason, like at the beginning of of your talk, you talked about like politics, and you were just talking about how you've got this doctor who's like, well, what if we did 7 days versus 14 days, and also kind of throw back to the important versus publishable versus fundable. Like, how do you, I guess, stay strong and navigate that landscape when like your findings might be revolutionary, but it's not worthy of being put in science, and you haven't gone through every single molecule in the supernatant and figured out what gene it targets or stuff like that? How do you like persevere?
Yeah, that's a hard one. That's what I was trying to say. It's obsession. I don't know. Why does the first chair violinist in the Cincinnati Symphony get to be first chair? Probably because he or she practiced all day long, slept with the violin. I don't know. I don't know how these things happen, but they do happen, and it does take a lot of perseverance. But how do you balance risk? There's a risk to your family, a risk to your health. There's a risk to your financial wealth. I mean, people make decisions. You're like, I'm even coaching my daughter. I have a daughter who's a urology resident at Duke in North Carolina. And, I'm like, you do whatever's good for your family. That's how the choice you make, because the rest is too hard. Her husband works, they have a kid, they have a dog. You want to make the best decision for you, for your career, but it might not be the best decision for your family. And so you say, how do I balance that risk? Well, I'm no, like I said, I don't give career advice, or I don't give relationship advice. So good luck.
Kind of just going off of that similar question you mentioned earlier, that you need whatever data you generate needs to be transformative to the field in order to get continuous funding. But a lot of time with exploration-driven science, you don't know what data you're going to end up with. So I was wondering if you had any advice on kind of just picking up on these things in the beginning.
These are great questions, and I don't know the answer. What would I remember watching the Steve Jobs movie with Ashton Kutcher was Steve Jobs in the first movie, not the second one. And the head of IBM comes up to him and goes, when are you going to get through your thick head that nobody ever is going to want a home computer? Nobody's ever going to want a personal computer. The head of IBM. And yet Jobs persisted. Elizabeth Blackburn won the Nobel Prize. Jennifer Doudna, you could say, well, they're both brilliant. They were brilliant, there's no question. But when your advisor comes up to you and goes, that's the stupidest idea I ever heard of, and then you win the Nobel Prize 30 years later, you can ask them what they were thinking when somebody said that to them There's a funny slide I've always seen. It says, everything is wrong until it becomes self-evident. Welcome to our world.
All right, thank you. So, oh, wait, we have one more? Yeah, we got one more.
Oh, yeah.
You're working on a project of?
With surgical adhesions, and I was curious if you had observed any data where you're seeing other surgical complications related to microbiome distortions.
Yeah, everything affects your microbiome, and your microbiome affects everything. So it's all involved. It's just another gene pool that you have to account for. We used to think, well, it's host genes. Like, Bert Vogelstein, who is a brilliant cancer biologist from Hopkins, he said, cancer is a genetic disorder. It was on the front page of Times, I remember it. It's genetic. Well, that turned out to be wrong. Okay, cancer is a gene-environment interaction. Well, no kidding. What is it? When you pass gas, it's a gene-environment interaction. So, yeah, people have shown that that microbiota, they use germ-free mice and scratch the intestine, and they don't get the same adhesions as if they have a conventional colonized mouse. So the microbiome plays a role in it. What role does it play? Don't beat up the bowel. We try not to. Do laparoscopic surgery. We try to when we can. But we can't control what that patient ate, whether they went to the Serengeti last year, whether they've been eating for breakfast, how many antibiotics they took the year before, or over their life. We can't control their life history. I guess what I'm saying is their life history is embedded in the ecologic memory of their microbiome and their host genes. So maybe someday in your lifetime, you'll get this readout of all the microbiota and all the host genes, and you'll say, oh, look, this signature portends a bad prognosis. Don't get a liver transplant. You're going to die. That's the hope. But we're a long way off from that.
All right, thank you so much for the wonderful talk, Dr. Alberti.